Basically, thank you very much, everyone, for joining. It's actually a pleasure to speak to you today, not only about science, but also a little bit about the career paths and decisions behind doing science. But I would actually like to start with some data and some projects, so you can see what I've been working on in the EV field in the last years. And then I'm happy to have a chat afterwards and discuss anything around those projects and how they came about, for instance. So the title of the talk, as Carolina said, is Fully Synthetic Extracellular Vesicles. So it's probably an approach to EV research that's quite unusual, but I hope I will be able to convince you that this is actually a valuable approach to follow in order to get some insights into extracellular vesicle biology. Please feel free to interrupt me at any time in this kind of informal setting if you any questions on the data or in the motivation behind this work comes up. First of all, I will be talking about two specific things today, two projects. One is actually has just been published 1 or 2 weeks ago in Advanced Science. And this project is about using synthetic extracellular vesicles to understand how the proteins on EVs actually come into work and how the vesicle underneath that protein plays a role in mediating its signaling functions. And the second topic I will be talking about is work that has been published last year in Science Advances. Those papers are not to be confused in Advanced Science and Science Advances. And that is more about the therapeutic functionality and possibilities using those fully synthetic extracellular vesicles. First of all, and that is maybe within the realm of this seminar series, I would like to introduce you to the field I did my PhD in actually, and that I've been working on, and this is bottom-up synthetic biology. That's kind of a new field within the life sciences. It's also somewhat originating from the physical sciences, but in general, it's an approach of biologists, physicists, and chemists that try to construct minimal life-like materials that should recapitulate and reconstitute some of cellular functions, but in vitro under well-defined conditions. So if we think about biological systems, either rather small-scale molecular machines like the ribosomes, we can in many cases appreciate that these structures have an inherent dynamic complexity inside. So we can describe them, we can image them, we have a model of them, but it's very hard for us to understand the dynamics behind because these systems are simply so complex, actually. So what we try to do in synthetic biology is to reduce the complexity of those systems. So create any kind of a system like those showing on the right side that will reconstitute cellular functions in a very defined environment, molecular defined environment. So what you see on the right side, those are microtubule esters forming within synthetic cells. So by doing this reduction in complexity, what we actually try to do is tackle some of the very fundamental questions in life. And that's a big picture behind the field. I'm not going to talk about these questions too much today, but as a general introduction into the field, things that we ask is where actually does matter become alive? So where's the transition from matter to life? How can we integrate living materials and synthetic materials into living structures? And how can we use those approaches to analyze the behavior of and the function of living matter? So, but today I will be talking about extracellular vesicles and basically in the interface of these two fields, synthetic biology and extracellular vesicle research. EVs, just to get clear on the definitions, is in that case anything that is a vesicular forms or an exosome, a microvesicle, oncosome, ectosomes, whatever. I don't think this needs much of an introduction in here. EVs are released by cells. They carry proteins and RNA inside, as well as other biomolecular contents. But today we'll be mostly focused on proteins. I will be not speaking too much about RNAs and RNA signaling mechanisms behind EVs. It's mostly about the protein signaling mechanisms inside. And I think tackling extracellular vesicles with synthetic biology is quite of a promising approach because also the EV field has a problem of very increased complexity. We know thousands of proteins and RNAs that we can find within vesicles. Vesicles themselves, EVs are very complex in their structure, they're very complex in their composition, and they're very diverse and complex within their function. And our job as EV researchers is to make sense out of that complexity, understand what is behind all those functionalities of EVs. So basically this interface between synthetic biology and extracellular vesicles is aimed towards the following project, which is the assembly, bottom-up assembly from molecular constituents of fully synthetic extracellular vesicles. On the one side, to mimic and recapitulate the therapeutic potential of synthetic extracellular vesicles, but on the other side also to study the molecular and functional signaling mechanisms behind. And doing this, we can circumvent some of the systematic problems associated to extracellular vesicles. We don't need purifications and isolation techniques like shown on the left side of this image, but we start from molecules, literally, so lipids, proteins, RNAs, and then we do some sophisticated biomaterial approaches in order to form structures that are very defined and very homogeneous in their function and structure, which we call synthetic extracellular vesicles. And then we analyze what happens if we interface such synthetic extracellular vesicles with cells in order to elucidate if they have a comparable therapeutic response, or in order to understand what is happening at the interface between an extracellular vesicle and a cell. So the goals of this is a systematic analysis of EV signaling, and by systematic, I mean like really check one by one in a very rational order things that are happening here. But also a second goal would be to assemble something that has the functionality, especially the therapeutic functionality of an extracellular vesicle population, but it is very safe for administration into patients because it's very defined and very homogeneous, very reliable, and so on. Now, the way we do this is on paper, more or less complex sequential process that happens in vitro from molecular constituents. I don't want to go into the details of this, but I can tell you it looks much more complex on paper than it is actually when doing it in the lab. So we have protocols published and open source to do this with basic standard lab equipment. The only thing you will need is a vortexer and some pipettes and an epi, basically, and the reagents for this, but those are very cheap. And essentially it involves, just to go over this very briefly, the two solutions, which one would be an aqueous solution that contains the lipids, the RNAs, and so on that you would like to have in the extracellular vesicles at the end, and an oil solution that will have a surfactant. And then you mix those two layers, those two phase separated layers by shaking, for instance, or by vortexing. What you will create are very tiny bubbles. You can do that also with sophisticated methods like microfluidics, and that thing will be very homogeneous, but you can do it with standard lab equipment. And in those bubbles, by controlling the chemistry of those surfactants, you will form extracellular vesicles that have all the RNAs that you put into the mixture basically encapsulated into that vesicle. And then in the third step, you remove all the oil again, and you go back into an aqueous environment, something that you can actually interface and incubate with cells. And at the end, you will simply couple the proteins that you would like to have in the extracellular vesicles on the lipid membrane in here. And you end up with something that has a similar structure, at least, like extracellular vesicles. So it's basically a sophisticated liposomal technology that is based on an extracellular vesicle blueprint, so a construction plan from extracellular vesicles. To give you an idea of how these things look like, and this is confocal imaging of the structures we produce, we have a lipid membrane. That lipid membrane can be coupled with proteins. In that case, that's CD9, a heavily used EV marker, and we have microRNAs in the inside. So the structure is basically the same as for natural extracellular vesicles. But just to highlight the definedness of the system, I'm putting here DLS-based size analysis of natural extracellular vesicles. And that's, I think, one of the results many of you get. You have different populations in an EV preparation. Well, if we do this with our fully synthetic EVs, the population that we produce is very much defined. We could discuss later on if that's a good thing or not. Biology is about diversity and heterogeneity, but if we try to understand something, sometimes maybe it's important to have something very defined and to kind of bury or introduce heterogeneity in a systematic way, step by step. But essentially, the important thing is that the structures that we produce, those FSCVs, fully synthetic extracellular vesicles, they can be incubated with cells, they're interacting with cells, they're uptaken by cells. And I will show you later on that also the cargo inside the lumen of those vesicles is released and functional within cells, but also the protein on the outsides play a role. So that's everything you need to understand. Don't try, so at this stage, it's not important to understand the way we produce these vesicle structures and so on. The important thing is what we produce looks like this and it has a functionality. We might argue and discuss to what extent it mimics the functionality of EVs, but in a certain way it does. So the first project I would like to introduce is a project that actually started, I think, in a very honest way, because I got into that extracellular vesicle field very naively. And that was one of the first questions that kind of came up. And it might be very simple, but I think many things that are simple need clarification also in the extracellular vesicle field. And that question was around, we know that cells can secrete soluble proteins, but we also know that proteins can be bound on extracellular vesicles. So why do cells come up with these two different types of releases? I'm not talking about RNAs in here. There's clear and very rational reasons to put an RNA into a vesicle that are kind of obvious, but for the protein, it's not directly clear. Why? What is the structural benefit of putting a protein on a vesicle? And that's one of the questions we were trying to ask in that project. Why do cells apply vesicular forms of protein, especially protein ligands and receptor ligands, and not their soluble forms in specific processes? And what is the role of the vesicle underneath those proteins? How does this contribute to the signaling and interactions of a ligand and a receptor? So we picked up to start with one model to work on, and that model is an immunological model that's basically relevant for the process when a T cell or any kind of cytotoxic cell attacks cancer or an infected cell. That's the process that's showing in this lattice light sheet microscopy image here. In that process, the two cells will form a very close contact, and it's known now for several decades that this contact is highly organized. It's something that we call an immunological synapse, and it involves a tiny cleft between the target cell and the immune cell. And in that cleft, we will find extracellular vesicles. So we know since the 80s that the attacking cell will release vesicles that are mostly referred as ectosomes into that cleft. And those vesicles carry proteins on top. And one of those proteins is the Phos ligand. It's also called CD95 ligand, for instance. And that Phos ligand is a cytotoxic protein, and it will induce the death of the target cell. Well, we choose this specific model mechanisms because I think it has some very distinct advantages for EV model systems as now known in the field. One is that the molecular signaling mechanisms of Phos ligand interactions and the downstream signaling, they're very, very well understood. We know what happens when the ligand binds to the receptor, which kind of reaction the receptor will need to go in order to initiate the apoptosis of the target cell. But also, we know from direct experimental evidence that EVs play a role in here. So we know that this ligand is secreted on EVs, but also in a soluble form. So this is why it's a good model system to analyze when is Phos ligand released in a soluble form and when is it released on a vesicle. And we know that this release is side-directed. In many projects in the EV field, we pick up EVs from cell culture media or from serum or whatever, and we don't know what cell released those vesicles and where did it release it to, where were they meant to go to. And in that system, we know it pretty clearly because there are two cells involved. One is secreting, one is the receiver. And at the end, we know that the soluble form, so the S Phos ligand, and the vesicular form, so the V Phos ligand, they have completely different cytotoxic potencies. So there's a real functional difference behind them. So we focused on this model system, and I think I need to introduce just the basics of what is important in Phos signaling here. As I said, there's a ligand involved and there's a receptor involved. And the ultimate goal of the ligand in this scenario is to induce hexamerization of the receptor. So it needs to bring 6 receptors in close contact because this will induce cross-phosphorylation and signal transduction into the cell. The ligand itself is a trimeric form. And what will happen if you use a soluble Phos ligand is that it will catch one receptor after the other until it's trimerized. And then 2 of those trimers will need to get into close contact to form a hexamer and induce apoptosis. That's the thing that will need to happen in the first place. Otherwise, there will be basically no apoptosis happening. So it's a multi-step process that involves multimerization of proteins. The system that we build now with our synthetic extracellular vesicles look basically like this. It's a vesicle, and we attached a recombinant form of the Phos ligand ectodomain by his-tag binding to those vesicles. The vesicles, as I said, can be very defined, and this will play a role later on in the project. But using this system basically enabled us to use the very same soluble and vesicle-based protein and compare them side by side. So we either used this recombinant Phos ligand in the soluble form, or we put this on a vesicle. So there's no biochemical difference between the soluble and the vesicle proteins that I will be showing you. If you're interested in how this looks like on the ultrastructural level, these are some cryo-EM images of such a vesicle with the proteins on top. And the first eye-opening experiment that we did is a very simple experiment. It measures the cell death by propylium iodide staining. And we compared either vesicular Phos ligand or soluble Phos ligand with some controls. And basically we saw that the moment we put the Phos ligand on a vesicle, it's cytotoxic, at least in that concentration. Soluble Phos ligand is also toxic in higher concentrations, but at this and I'm talking about picograms per ml in this case, we can induce significant cell death simply by immobilizing a protein ligand on a vesicle. And I think that's a very important and very fundamental, maybe too simple observation, but it's very important for the EV field that proteins immobilized on vesicles can have a very distinct functional characteristic compared to their soluble forms. So we ask, why is that the question? We made tons of controls, that it's not about the vesicle concentration, that it's not about density, that it's not about the way, the orientation, and so on. And it basically, every time it came out the same thing. Put this recombinant protein on a vesicle and it will be cytotoxic. So in order to kind of clear our minds behind that, we did some diffusion simulations in here. And the basic idea behind those simulations is shown here. We used in kind of a model for an immunological synapse, either the soluble forms of the Phos ligand shown in blue or vesicle-bound forms of those Phos ligand. And we kind of just push them into that diffusion space and let them interact with a simulated membrane full of Phos receptors that were either, and we track basically the hexamerization events. So how many times does Phos ligand, either soluble or vesicle bound, attach to the membrane, bind to a receptor, bind to 2, bind to 3, bind to in order to form a hexamer and so on. And we compare this over time. And what we find in accordance to our experiments is that the vesicular form is way more potent in inducing hexamerization events. That's what's plotted in here over time compared to the soluble form. And the reason for this is not, as we initially thought, that simply the local concentration is higher. If you put it on a vesicle, you basically increase the local concentration of Phos ligand on the membrane. it's a completely different aspect. And I think it's a potentially universal aspect behind EV signaling. And we call this effect the vesicle-induced receptor sequestration, the VIRS effect. So VIRS came out from those simulations. And it basically is a very simple mechanism. And because it's simple, it could be universal. When we compare the soluble form of that signaling stage, we could imagine ligands that have bound two receptors, ligands that have one receptor, and hexameric ligand receptor complexes. But these two will need to find each other by diffusion over this long and large membrane area. In contrast, if we have the ligands on a vesicle, what will happen is that their ligands will bind to the receptors, and the receptors could leave that vesicle binding area, but the ligands cannot because they're immobilized on the vesicle. So we will up-concentrate the receptors at the vesicle attachment sites. And by this, we will foster, in this case, hexamerization, but in general, receptor-receptor interactions. So basically, we increase and we sequester step-by-step receptors at that binding site of the vesicle to the target cell membrane. And in order, so this was a model, a hypothesis, and we set out by using our fully defined vesicles to test it and to verify that this is actually the most prominent process in play, and the explanation why this is actually happening, that the proteins are so much more potent when put on a vesicle. And one of the predictions of that simple model is that when you use a large vesicle and we use a smaller vesicle, if you keep the amount of ligands per vesicle constant, the hexamerization should be increased because that vesicle binding area is smaller. For a smaller vesicle, the sequestration area is smaller. So that's basically what we find in the simulation. This is simulation data for 3 different radii of vesicles. But if we do now the experiment and we produce differently sized synthetic EVs, we can see the same trend. First one, so this is again measuring cell death, untreated controls, and soluble fast ligand. There's basically no difference. But if we now make the vesicles smaller and smaller and smaller, we can actually increase their cytotoxic potency. And we know, based on different controls, this is not because of different diffusive properties of the vesicles or different binding and stochastic and so on. That's really because of this. But I also want to show you a second proof of that model, which is basically focused to show that there's a real sequestration happening in terms of the receptors cannot leave that area. And for this, we turned it to a microscopy analysis based on GFP-tagged Phos receptors. So in that case, the receptor in the target cell is fluorescently labeled, and we can image the localization of Phos, so the receptor in the target cell membrane, and we can co-localize this localization with the vesicles. So you can see here in orange vesicles attached to a target cell membrane. Those vesicles have Phos ligand on top. And at the sites of vesicle attachment, we see increased fluorescence of the Phos receptor. Well, that's something you would, kind of expect, because if you have a ligand, maybe the receptor will be around. But the question is, can the receptor actually leave that place, or is there a sequestration taking place? And for this, we did fluorescent recovery after photobleaching analysis, FRAP. So we bleached away the receptors and we watched the recovery. Are new receptors coming in and old ones basically leaving? And if we do this, this is the condition shown for a control. So we basically are able to bleach the receptors. If we do this for free receptors, we can see a nice recovery, as you would expect. Fresh receptors are coming into the bleached area. But at the sites of vesicle attachment, there's no recovery. So old receptors can basically not leave that area. And there's a real sequestration mechanism taking place here. So I showed you now these two kind of approaches to verify this vesicle-induced receptor sequestration. One, that vesicle size plays a model, which is plays a role, which is a prediction, and the sequestration is actually taking place here. So to conclude this part of the project and presentations, of basically the paper behind, which is the VIRS paper, we think that there's this general mechanism playing a role for extracellular vesicles, a site-directed multimerization based on the vesicle itself. So we think, and I showed you this now for Phos ligand. In the paper, we also report on RUNC-L ligands, but I think there's many other receptor ligand interactions where that might play a role. We could think about interleukins that are released in soluble and EV-bound forms. We could think about Wnt receptors, Wnt signaling, where we know that there are Wnt ligands on vesicles, but also in a soluble form. We could think about all the EGF, growth factors, where we have the same scenario. In all these scenarios, the receptor multimerization and interactions play a role. So I think this is a potentially universal mechanism in here. Also, I showed you that simply taking the ligand, and I still like that observation somehow, simply taking a protein ligand, put it on a vesicle, it will increase and really dramatically increase its potential. From measurements, we estimate it's between 100 to 1,000 times an increase in the signaling potential. So essentially, we think that EVs are there to promote the accumulation of receptors in the membrane plane and the membrane area, and thereby foster cis interactions. And in that view, extracellular vesicles can be somewhat considered as a concentration device. And you could think about this more as maybe a satellite system where a cell kind of needs to provide an interface to present a protein to a long distance cell, maybe. So two cells that cannot touch but still need an interface in order to signal. That's the scenario where maybe extracellular vesicle presented proteins could be playing a very, very important role. And at the end, I think to answer the question, the structural profit and the why is that EVs are kind of a template that will promote the outside-in signaling based on self-organization mechanisms in the first instance. So this is concluding from the first paper, the very recent paper that came out. But I also want to show you the first, our initial project that we started with, and that explores the therapeutic functionality of those synthetic extracellular vesicles. I think it's pretty clear that the moment we understand a biological system, we can also, most cases, apply it for therapeutic functionality, at least in a human-based system. And that's obviously something that we strive as researchers and as scientists. So obviously, extracellular vesicles are important for diagnostics. They're mirroring the physiological states of the cells of origin, and we can recover them quite easily. So liquid biopsy is a hot topic here, but also EVs are good candidates for therapy because they're endogenous regulators of physiological states, and we can purify EVs and re-administer them back into patients for a therapeutic purpose. The limitations are pretty clear for all of you, I think. We have the production conditions are fluctuating, it's time consuming, and it's somewhat error prone. And again, I want to come back to the complexity problem. It's because we have limited quantitative understanding of this, but we have also limited engineering capabilities of extracellular vesicles. So the top-down design properties of that system are somewhat limited. And here, bottom-up comes into play. And I want to show you one therapeutic example that we've been following up with our extracellular vesicles. And this one is based on a wound healing model, actually. So it's a regenerative medicine approach. I will show you wound healing data. The paper also contains some vascular regeneration data. So basically where we try to induce and promote neovascularization of tissues, but this will be mostly now focused on wound healing. So having that tool in mind of being able to produce extracellular vesicles, what we need now is actually the blueprint that I was talking about. We need the construction plan, right? And that's when we turned back into literature and looked at what's up there, what has been published, and we came up with that paper from Geiger and colleagues that describe fibrocyte-derived exosomes that would accelerate wound healing in diabetic mice. That paper, the authors described three very prominent effects. First, that epithelial cells treated with those exosomes increase their proliferation. Second, that they migrate faster. So two things you would expect for wound healing. And then also that they remodel extracellular matrix, so the collagen deposition is increased here. And they describe some of the molecular components of those exosomes. Those are the typical tetraspanine, CD9, 63, and 81, some of the microRNAs present, and then we know that the lipid composition, the typical lipid composition of EVs. And that's what we did. We built basically these extracellular vesicles. So we employed our system in order to build fully synthetic extracellular vesicles for wound healing therapy. And I want to show you one of those in vitro assessments that we did in order to analyze the functionality of our synthetic extracellular vesicles. And those are in vitro scratch assays for wound healing. So basically you culture human epithelial culture, and then you introduce a wound in there, and you just watch how those epithelial cells will close the wound. And you can measure this basically by measuring, you can quantify this by measuring the migrated area. And here, one of the, I think, very elegant advantages of the bottom-up approach come into play. Now we can really test the effect of every single component of the tetraspanines, of the microRNAs, and see what is their functional contribution in here. So we saw that already just putting the proteins on top will kind of improve that migration and healing in that project. And then adding on top some microRNAs will further increase it until we almost get full recovery of that healing site. So we concluded from this that our fully synthetic extracellular vesicles are actually able to close at least artificial wounds of keratinocytes in vitro. We did, or we found the same thing also for the ECM remodeling. We found the same thing for the proliferation increase, so those phenotypic effects that I showed you in the last slide. So all that was present in vitro, in vitro models. But we wanted to take this also a step further and see, does it really perform on skin models actually, so in a real system. And for this, we used organotypic human skin models that were basically introduced with the epithelial wound shown up in here. So those are histological sections. And that wound site was either treated with the human serum as a positive control, a buffer, or our fully synthetic extracellular vesicles. And what we measured is over time, that is now here after 48 hours, I think, the wound diameter, so how much has it closed, and the thickness of the epithelium that was regenerated here. And as you can see, and we can resolve this again for the individual components, the fully synthetic extracellular vesicles really dramatically improve and boosted that healing process, just as Geiger and colleagues described in their paper for the real extracellular vesicles. And I think this kind of nicely demonstrates that you can take a biological blueprint of an extracellular vesicle. If you know the crucial components, we can now introduce them into a synthetic system that is very homogeneous and very safe to use. And further on, and I'm not showing you this here, but it's in the paper, you can then quantitatively tune the composition. You can put in more of, microRNA A and then a little bit less of microRNA B and kind of fine tune the functionality. And we show data in the paper that this is actually happening, that there are optima for different components that you need to introduce. So it's once you have established it, it's a very elegant, fast, and reliable system to exploit the functionality of EVs for therapy. Just a last slide of data for this talk now. You can also use this not as a functional system, but you can also use this if you're interested in uncovering the mechanisms behind that signaling processes. For instance, by combining fully synthetic extracellular vesicles with RNA sequencing. So a very simple experiment that I'm showing you here is taking those fully synthetic extracellular vesicles I showed you, incubate them with keratinocytes, and then do RNA sequencing. You will find differentially expressed genes, and you can pinpoint the pathways that have actually been triggered by the incubation with those vesicles. You will find pathways that will not surprise you if you know the functional data, for instance, that everything related to proliferation is increased, everything related to matrix remodeling, migration, cell-cell interactions, that's upregulated and actually tuned by the extracellular vesicles. But now even more important, so this you could also do with natural extracellular vesicles, right? You could take your preparation, incubate it. But now you can again do the sequential step, and in this case I'm showing you for the proteins, and sequentially check which kind of a protein is actually relevant to trigger what kind of a pathway. I just want to highlight the combination of CD9 and CD81 that's really responsible to trigger all the immune associated processes behind the wound healing in here. And that's kind of an additive process. And with that kind of experiment, you can sort out, okay, I need these two components to trigger something at least. Yeah, you can do it, and we show in the paper, you can do that also with the microRNAs. Then it becomes a problem of pipetting. How much pipetting are you willing to do, and how many conditions are you willing to test? And then obviously a problem of money. How much sequencing can you do? But the fundamentals of this can be applied for every system at the end. And I think this is a real improvement and a real advantage tool that can be provided for the EV community in this case. So just to conclude on this side, this is a proof of concept that synthetic extracellular vesicles with therapeutic functionality can be designed and assembled in a systematic way. We can identify key microRNA and protein compositions that are underneath that signaling. And by this, fully synthetic extracellular vesicles kind of overcome some of the natural limitations of natural extracellular vesicles, because we overcome the complexity and the molecular diversity behind the system. I'd like to thank all the people that are involved in that work. That's mainly Joachim Spatz and Elia Platzmann that were leading this research at Max Planck Institute for Medical Research. Obviously, all of the co-authors of the paper that were very important to get this work done. So that's not all my work. I want to highlight Franziska Dietrich, who did the human skin model work. I want to highlight Julius Fischler, who did the modeling work, for instance, many collaboration partners. But obviously, I would also like to thank the organizers of the seminar series for inviting me to present. And I really also want to highlight that I think this is a very important thing, because I've talked to many master students, PhD students. I know it from myself. You get lost in translation in that case. Yeah, you get lost in the scientific system. It's important to provide platforms where we can discuss about what's a good decision, what's a bad decision to do, what you should be taking care of. And I'm happy to discuss now the data. Obviously, I'm always looking for input on the data, but I hope we can also have a discussion around how one can find a path in academia. I need to say I'm in my 1.5-ish postdoc now. So I'm not a senior professor that could give you a career path decision, but I could serve maybe as one example for you to see what I did and what were my reasons behind. And you can either use that information for yourself or use it as an inspiration, or maybe I'm also a good negative example for you guys. That's this up to you. But first of all, thank you very much for that attention. And now I'm looking forward for a discussion.
Thank you very much, Oskar. It was a really nice presentation, and I'm really happy if there are questions, and also questions related, as you mentioned, on the careers and everything, because it really is important to share experience and to try to suggest the different different paths to everyone. Yeah. I'm really happy. If there are questions, please, you can unmute yourself and ask directly to Oskar. Hi, David, please.
Yes, hi, Carolina. Thank you, Oskar. Amazing talk, very nice data. And I agree, it's a great platform. I was wondering, what do you think those vesicle-bound ligands or those synthetic vesicles, do you think they would also work to bring T cells and cancer cells together, like similar to bispecific antibodies, just more stable, longer half-life?
Yeah. That's a great question. So actually, it's a great question because it's a good scientific question, but it's also a good question for that format, because turns out I'm now working on bispecific antibodies in my postdoc. So based on that, I know that EVs and other kind of materials are kind of explored to serve as a platform to tune the regulation of that interaction. Yeah. So can we put ligands for a target cell and for a T cell on a vesicle, and by this force these two cells together.
Exactly.
And I think there's a good amount of data that these kind of approaches work. I know that vesicles and liposomes have been tested for this, and I don't see a reason why our system could not be used in the same way. We simply did not do this at the moment, but because it's a defined system, I think it's attractive because you can, you know, in that interaction, immune interactions, it's not about on and off, it's about tuning, you know, fine tuning and regulating how much you want to activate a T cell, how much you want to direct it towards a cancer cell or towards an antigen. And if you have a platform that you can engineer, talking about size, charge, fluidity of that vesicle and so on, that might provide you a functionality that's easy to tune and potentially very powerful. So I would say yes, yeah, that could be applied.
Sounds good. Yeah, looking forward to your next paper then.
Thank you.
Okay, thank you. There are other questions? Yeah, please.
Yeah, thanks, Oskar, for this really nice presentation. I think it's a really fascinating topic to produce this fully synthetic extracellular vesicles. I was wondering, how do you determine the lipid composition of your vesicles? Is this based on literature?
Yes, essentially, yes. It's so there's, I guess you guys know lots of work on identification of components based on RNAs and proteins out there. And there's comparably less literature on quantitative measurements on the lipid composition of EVs. We did not do measurements of those wound-healing EVs ourselves in terms of the lipid composition, because I think the methods, it's mostly mass spec, what you will apply for this, that you will need are very sophisticated. And you need very good calibration and a lot of know-how in order to do very good and reliable experiments and quantifications in here. But the data banks that are online can provide you with this information of the lipid composition, because EVs, well, let's say, originate from a restricted number of pathways, let's say either from the plasma membrane or intracellularly. The variation in terms of the lipid composition are usually not as huge as the variations, for instance, in the RNA composition. That might dramatically vary between cell types and so on. So the typical lipid components that you will find in EVs are relatively stable, although their ratios might vary between cell types, for instance. But we can change those compositions and we can test in the same way as I've been showing you for the proteins and RNAs, how much does it play a role? But this is in most instances not a direct functional role. So we have lipids like phosphatidylserine that are actually receptor ligands and will induce immunological effects. But most of the lipids come to play when it comes to the biophysical properties of the vesicle, fluidity, charge, and so on. And we've started to look into what role does the lipid composition play of those vesicles. So again, short question, yes, we take it from literature.
Right, thanks.
Okay, there are other questions. Meanwhile, I can ask you one, but it's just a curiosity. How you can determine the size of your synthetic vesicles? Is it a matter of shaking? Is a matter of different oil?
Yeah, so basically, if you do this by microfluidics, you can obviously tune it by tuning the microfluidic system. If you do it by shaking, you will determine it by the duration of shaking and how hard you shake or how hard you vortex, basically. And what you will need to define is the size of those droplets that you generate. The size of the droplet will determine the size of the vesicle at the end.
Yeah, but the oil that you use is always the same, or you also change the oil for changing the?
It's always the same. If you talk about orders of magnitude, so you can use the same system to produce vesicles that are cell size. 50 microns or whatever. Yeah, because you might want to build a synthetic cell. If you want to produce this, you might need a different surfactant composition compared to if you want to build a vesicle that has 300 nanometers and should mimic an EV at the end. But that is not a lot of variation that you will need. And in general, it's a constant system on the old side. Okay, thank you. And another question regarding the data that you show us with the protein and the higher functionality of the vesicles associated protein. Do you think that this is something that can happen also in a let's say more easy system in which you have one protein and one receptor and not a clusterization of the receptors? So, yes, I think so. So the concept is in that sense also universal and translational to other protein systems, because well, so most receptor interactions require cross-phosphorylation, for instance, or receptor multimerization in any kind of a sense, so where you don't need the first trimerization step. But almost no ligand receptor activation involves only the ligand and the receptor. There's usually always co-receptors used. Let's think about T-cell receptors, for instance, that are activated by MHC complexes. You will need basically the binding of the MHC complex, but you will also need co-receptors on the side, CE3, for instance, and so on. So if you have a point on this huge plasma membrane where all things can come together and stay together for some time, then that system can be applied also for proteins and ligands that don't need trimerization and hexamerization behind that. Okay, thank you. Other questions from the audience? Okay, so if not, I really want to thank again you, Oskar. It was really nice. Thanks for your presentation, and thank you everyone for following us, and thank you again. Bye, everyone. Bye-bye.
Bye.